Okay, I think we can start. Hello, everyone, and welcome to this lecture of the computer architecture course. Today, we are going to have 4 interesting talks. They are from papers from our group, and they are very timely because the 4 of them belong to the micro conference that is starting this weekend. The first paper, GenASM, will be presented by Damla, who is the first author. This is an accepted paper at Micro this year, and she will be presenting it next week. I think it's on Monday. This is a collaboration of CMU, where Damla is doing her PhD, Intel, and ETH. And Damla will present the GenASM, which is an acceleration framework for genome analysis. And it's the first near memory hardware accelerator for the Bitap algorithm, which is used for approximate stream matching. When you're ready, Damla, you can start.
Okay.
Thank you very much.
Thank you so much for the introduction, Juan. So hello all, and as Juan already introduced, I'm Damla. I'm a PhD candidate in the Software Research Group at Carnegie Mellon, advised by Professor Onur Mutlu and Professor Sagata Goz. And today I will be presenting our work, GenASM, which will appear in Micro 2020 next week. So GenASM is a high-performance, low-power approximate string matching acceleration framework for genome sequence analysis. So genome sequencing, which enables us to determine the DNA sequence of an organism, plays a pivotal role in areas such as personalized medicine, outbreak tracing like COVID-19, and the understanding of evolution. Modern genome sequencing machines extract small randomized fragments of the original DNA sequence, which we call reads. And state-of-the-art sequencing machines produce either short reads or long reads, and this is based on the length of those DNA, like part of the DNA sequence. And for short reads, we observe a very low error rate, which is around 0.1% of the length of the reads. And for long reads, which we can see thousands to millions of base pairs in each read, the error rate is relatively much higher compared to the short reads, and we observe 10 to 15% of the read length for the error rate. So read mapping is the first key step in genome sequence analysis, which we will go deeper, but you should be familiar with the term from Professor Onur's or yesterday from Mohammed's talk. So in read mapping, what we do is we take each read, align it to one or more possible locations within the reference genome, and find the matches and differences between the read and the reference genome segment and that location. So multiple steps of read mapping must account for the sequencing errors, which I have already introduced, and also for the differences caused by genetic variations. As you can imagine, although we share more than 99% of our genome, we all are different in terms of appearance or the genetic features we have. So these are caused by genetic variations. And as a result, read mapping must perform approximate string matching or ASM in order to consider or take account for these type of errors and differences when performing read mapping. So it is currently bottlenecked. Of course, there should be a downside, right? By the computational power and memory bandwidth limitations of existing systems. And by the way, you can interrupt me at any point. You don't need to wait for the until I finish my presentation. So our goal in this work is to design a fast and flexible framework for both short and long reads, so that we should support both scenarios, which can accelerate multiple steps of genome sequence analysis. So to this end, we propose GenASM, and we base GenASM on Bitap, which is an approximate string matching algorithm that uses only fast and simple bitwise operations, which makes it amenable to efficient hardware acceleration. And to our knowledge, GenASM is the first work that enhances and accelerates Bitap. So we modified Bitap to support long reads and to enable parallelization. And we also develop a novel Bitap compatible algorithm for traceback, which we will come to the details. And then we also co-designed specialized hardware for both algorithms, for modified Bitap and the novel traceback algorithm. So GenASM is flexible and can be used to accelerate multiple use cases, which involves approximate string matching, as you can imagine. And in this work, we evaluate 3 use cases in detail, from genome sequence analysis, read alignment, pre-alignment filtering, and edit distance calculation. So if you attend yesterday's talk by Mohammed, you should be familiar with these use cases, but I will explain them later in detail. So we find that for all these 3 use cases, GenASM is significantly more efficient in terms of both speed and power consumption than state-of-the-art software and hardware baselines. Okay, this was the introduction of GenASM. So let's look at genome sequencing and sequence analysis, and also approximate string matching and Bitap algorithm in detail. So as I have already introduced, the goal in genome sequencing is to determine the order of the DNA sequence. In other words, the order of ACGTs, which is the characters that you would expect in a DNA sequence in an organism's genome. However, so these sequencing machines cannot take that long DNA as an input. And by long, for example, for human genome, I mean 3.1 billion characters or base pairs. And the machines cannot take them as a whole and give the complete sequence as output. Instead, all sequencing machines chop this DNA into pieces and only identify relatively small pieces. And these small pieces lose their location information with respect to the original genome. So we don't know how they fit together. We lose that information. To see it with an animation, think about this as a large DNA molecule. And as I said, it can be 3 billion characters for a human genome. And the whole genome cannot be sequenced all at once. So we break it into smaller fragments. And then we sequence each of these smaller fragments, and small pieces of DNA sequences, as I said, which are called reads, are generated. So why genome sequencing is important? And I will provide an example from this year, and which is very relevant because whole world is experiencing this right now, which is COVID-19. So why genome sequencing and sequence data analysis are important. So after, as I said, after genome sequencing, we obtain a data, but we need to analyze it. And after this analysis, what we can find out, or why it's important, it is important to detect the virus, for example, from a human sample like saliva. Or it is important to understand the sources on modes of transmission of the virus. Or to sequence the genome of the virus itself, for example, here in COVID-19, in order to track the mutations in the virus, which would help to understand also the sources and modes of transmission of the virus. So if you sequence the virus from different locations and compare the outcome, then you can see for each location, or for some portions of the location, where that virus transmitted from. So you can see, okay, for example, in Washington, DC, the virus came from Europe. I'm just making it up, but you can track it, and it is important to see the transmission. And also to explore the genes of infected patients. So this is not for sequencing the virus, but sequencing the human itself, to understand why some people get more severe symptoms than others, and also to help with the development of new treatments like vaccines. So sequencing of COVID-19 here is pretty important to address these requirements, to both understand the virus and to come up with solutions to defeat that. And what we should expect in near future, I'm saying near future because there's already a development, like a sequencing device development happening in a company, sequencing technology company called Oxford Nanopore. So as you can see from the right-hand side, they are introducing a very small, portable, and cheap devices that can be plugged to your mobile phone, which would enable everyone to perform sequencing, like genome sequencing, on their own genome by themselves using their only mobile phone. So this would provide portability, easy access, and also privacy as well, because you can do it by yourself. But challenges, of course, as I said, these devices are generating some data, but we need to perform the required analysis on our phones, which have very limited resources. So what do I mean when I say genome sequence analysis or genome analysis? So this is this slide should be familiar because this is one of the Professor Onur Mutlu's talks. So after sequencing, as I have already introduced, we need to find the location of each of these small pieces with respect to a reference genome. And by reference genome, what I mean is, for example, we have this for many, many organisms, including human genome. It is a representative like the DNA sequence. So of course, it is only generated from a sample, so it cannot be representative for the whole, like all organisms. As I already said, we have some differences, but this is the approach we need to follow, at least to find like the location of those small chunks, because that's how you can then find the differences or the matches with respect to this original genome. After performing this, the next step is, of course, seeing what are the, based on the differences we discovered, calling the variances or any diseases, for example, or any genetic features that are mapped to those differences. And then the last step is, of course, the scientific discovery based on this variant calling step. So again, this is another slide from Professor Onur Mutlu. Here, so it is known that the bottleneck is in the read mapping step. So that's why in GenASM, we mainly focusing on that bottleneck. So let's focus on read mapping and see what's happening as part of its pipeline. So it can be seen as a 4-step process, and sometimes it's also called seed and extend. And first, read mapping starts with indexing the reference genome. And this index contains all possible fixed length substrings, which we call seeds, as the keys, and the exact match locations of these substrings in the reference genome as values. So we come up with a hash table, and here we have the keys as like substrings, and they are fixed length, and their exact match locations in the as the values. And then we in the seeding, we queries this index structure for determining potential mapping locations of each read in the reference genome, and using the substrings of the reads, which are again called seeds from each read. So that's why it's called seeding, because we are querying that generated hash table based index with the seeds coming out of the reads. And seeds are, as I said, the substrings of the read. And in the third step, pre-alignment filtering, we use filtering heuristics to examine the similarity between every read and its potential matching segment on the reference genome that are identified in the second step. And here, goal is to eliminate most of the dissimilar sequences to decrease the number of required alignments, which is the next step. So here, we provide the remaining potential mapping locations after we apply the filtering heuristics. And then for the non-like filtered remaining mapping locations, we get the reference segment and query read in the fourth step, and perform read alignment, or also called seed extension, to, which performs a dynamic programming-based algorithm to determine the best location of the query read and to find the optimal alignment. So when we are mapping the reads generated from the sequence genome, they may not exactly map to the reference genome, as we know, because of variations and sequencing errors. And let's look at this example to see what kind of errors and differences, which are also known as edits, we should expect. So first, we can observe deletions. As you can see here, one of the characters of the reference is missing in the read, so it's deletion. Or we can expect substitutions, which means that one of the characters in the reference flips with another one. Here, for example, G is not existing, but instead we see a C there. And the other option is seeing insertions, which means that a character that doesn't appear in the reference can be introduced in the read. So these are the three common errors or edits we can expect. So since multiple steps of read mapping must account for these errors, deletion, substitutions, insertions, they must perform approximate string matching. And the goal of approximate string matching is to detect the differences and similarities between two sequences. And in genomics, ASM is required first to determine the number of, a minimum number of edits between two genomic sequences, which is called edit distance. And here in read mapping, the reference segment and the read itself are those two genomic sequences. And also we need ASM to find the optimal alignment with a traceback step. So in this traceback step, what we do is we find the sequence of matches, substitutions, insertions, and deletions along with their positions. So for example, when we look at this example here, we see that we have 3 matches followed by a deletion, and then 6 more matches and a substitution, and 6 more matches and an insertion and 2 matches for this example. So this is the traceback output we want to come up with. But approximate string matching algorithms need to perform this additional traceback step in order to find that, because as the first step, they only find the edit distance, or in some other scenarios, the score of the alignment. So we need to do this additional step to come up with this output. And this is revealing the location of each edit, as I said, and type of each edit, which you should expect in the alignment, in the optimal alignment. So, excuse me. Okay. So approximate string matching is typically implemented as a dynamic programming-based algorithm, which has quadratic time and space complexity. So it is desirable to find lower complexity algorithms for approximate string matching. One candidate to replace DP-based algorithms for ASM is the Bitap algorithm. And Bitap uses only fast and simple bitwise operations to perform approximate string matching, which makes it amenable to efficient hardware acceleration. And here, that's the reason why we picked Bitap algorithm to accelerate. So Bitap tackles the problem of computing the minimum edit distance between a reference text, such as reference genome, and a pattern, such as a read, with a maximum of k errors. So k is a parameter here. And the algorithm has 2 steps. And it starts with a pre-processing procedure where we generate one pattern bitmask for each character in the alphabet, which is ACGT in this context. And these pattern bitmasks help us to represent the query pattern in a binary format. And the second step, searching or edit distance calculation, which is the main step of the algorithm. And here, we examine each text character one by one by using the pre-processed pattern bitmasks from the previous step, and status bit vectors, which are updated at each text iteration and hold the partial match information for the text characters examined so far, and simple bitwise operations. So let's look at quickly the main step of the algorithm, edit distance calculation. As I said, the algorithm examines each text character one by one, one per iteration. And at each iteration, the pattern bitmask of the current text character, which is denoted as PM here, is retrieved. And then for a distance D, which can be between 1 to K, and K was the, if you remember, the like threshold of edit distance, which is a parameter of the algorithm, 3 intermediate bit vectors for the error cases, one each for deletion, insertion, and substitution, and another one for the match case are calculated. And in order to take all possible partial matches into consideration, which are stored with these 4 intermediate bit vectors, we perform an AND operation with the computed intermediate bit vectors to preserve all zeros that exist in any of them. And in this algorithm, zeros means match, so that's why we care zeros instead of ones in this as part of this algorithm. And we save the ended result as the RD, which is that status bit vector I mentioned in the previous slide. And it is the status bit vector for the current iteration. And this process is repeated for each potential edit distance value, as I said, from 1 to K. So this is an existing algorithm. We haven't like developed that. But why I'm explaining this? Because it is important to understand 3 key properties of the algorithm which shaped our design actually in GenASM. So first, Bitap iterates over each text character as the outer loop, as I showed, and it iterates over each possible edit distance, 1 to K, as the inner loop, again, as I introduced. So when we have long error-prone reads, both of these loops need to iterate over very large numbers. And this is for each read to reference mapping alignment. So it is huge. We are dealing with large number of iterations here. Second, there's two-level data dependency in Bitap. First, there's an outer loop data dependency when computing deletion, substitution, and match bit vectors, as they require OLDR bit vectors, which are coming from previous text iteration. And also, there's an inner loop data dependency when computing insertion bit vector, as it requires RD-1, which is coming from previous inner loop iteration. So we are dealing with two-level data dependency. And third, Bitap generates these 4 intermediate bit vectors, but they are not stored and processed with a traceback step to find the optimal alignment. So there is no traceback support in the baseline Bitap algorithm. So we find that Bitap has 5 limitations that hinder its applicability and efficient hardware acceleration for genome sequence analysis. And we have already covered the first 2 algorithmic limitations. And we have one more algorithmic limitation, which is no support for long reads. And in Bitap, the query length, so read length, is limited by the word size of the machine running the algorithm, because each bit vector has a length equal to the length of the pattern or read, and we perform the bitwise operations on these bit vectors. So the algorithm doesn't support long reads whose lengths are on the order of thousands to millions of base pairs, because we don't have the word limit, is the word size is limiting us. And these are the algorithmic limitations of the Bitap algorithm. So even after we solve the algorithmic limitations of Bitap, we find that we cannot extract significant performance benefits with algorithmic enhancements alone. Excuse me. Okay. For example, while Bitap iterates over each character of the input text sequentially, we can enable text-level parallelism to improve its performance. However, the achievable level of parallelism is limited by the number of compute units in existing systems. Then we would expect that a GPU, which has thousands of compute units, can overcome the limited compute parallelism that CPUs experience. However, we find that a GPU implementation of the Bitap algorithm suffers from the limited amount of memory bandwidth available for each GPU thread. And these are the hardware-related limitations of Bitap. So in order to overcome all of these limitations and design an efficient accelerator, we find that we need to both modify and extend the Bitap algorithm and develop specialized hardware that can exploit the new opportunities that our algorithmic modifications provide. To this end, we propose GenASM, an ASM acceleration framework for genome sequence analysis. So to our knowledge, GenASM is the first ASM acceleration framework for genome sequence analysis, and it is also first that enhances and accelerates Bitap. So we make significant modifications to Bitap in order to overcome those 5 limitations. First, we modify the Bitap algorithm by extending it to support long reads and by eliminating loop carry data dependencies to enable parallelization. And second, we develop a novel Bitap compatible algorithm for traceback, which uses information collected during ASM about different types of errors, which I mean those intermediate bit vectors, to identify the optimal alignment of reads. And these cover the algorithmic contributions of GenASM. And third, we co-design specialized low power and area efficient hardware for both modified Bitap algorithm and the novel traceback algorithm, and this covers the hardware level contributions of GenASM. So our first algorithm, GenASM-DC, is the modified Bitap for distance calculation, where we extend it, as I said, to provide efficient support for long reads, and also we enable parallelism besides bit parallelism that baseline Bitap already has by performing loop unrolling and removing the loop dependencies, and also exploiting text level parallelism, where we divide the text into overlapping subtext and search the query in each of these subtexts in parallel. And our second algorithm, GenASM-TB, is a novel Bitap compatible traceback algorithm, as I have already introduced, which walks through the intermediate bit vectors, match, deletion, substitution, and insertion generated by GenASM-DC, which is the modified Bitap, as I said, and finds the optimal alignment. In order to decrease the memory footprint of the algorithm, and this is coming from storing all these intermediate bit vectors for each inner and outer loop iteration. So it is huge. It follows a divide and conquer approach. And here we divide the text and pattern into overlapping windows and perform the traceback computation for each window. After all of the windows' partial traceback outputs are generated, we merge them to find a complete traceback output. So this helps us to, since we are dividing the text and pattern, decrease the amount of data we need to store as the intermediate bit vectors to perform traceback. So let's look at the hardware design of GenASM. Our co-designed hardware consists of two components. And GenASM-DC here provides hardware support to efficiently execute our GenASM-DC algorithm to perform distance calculation. And GenASM-TB, which provides hardware support to efficiently execute our novel GenASM-TB algorithm to find the optimal alignment. And GenASM has two types of SRAM buffers, DC SRAM and TB SRAMs. And let's look at the high-level overview of GenASM. So GenASM execution starts when the host CPU issues a task to GenASM with the reference and query sequences locations, and then GenASM-DC reads the corresponding text and pattern from the memory, and then writes these to its dedicated SRAM, which is DC SRAM. After that, GenASM-DC divides the reference text and query pattern into multiple overlapping windows, and for each subtext and subpattern, which corresponds to each of these windows, for each subtext and subpattern, GenASM-DC searches for the pattern within the text, like subpattern within the subtext, and generates the bit vectors. And then GenASM-DC writes these bit vectors to TB SRAMs. And once GenASM-DC completes its search for the current window, so after it writes all of the bit vectors for the current window, then GenASM-TB starts reading the stored bit vectors by following a input dependent order, and then it generates the Windows traceback output. So once GenASM-TB generates this output for the current window, then we compute the next window and repeat steps from 3 to 7 until all windows are completed. So our specialized compute units, which is DC and TB accelerators, and on-chip SRAMs, which are DC SRAM and TB SRAMs, help us to match the rate of computation with memory capacity and bandwidth, achieve high performance and power efficiency, and scale linearly in performance with the number of parallel compute units that we add to the system. Let's look at GenASM-DC's hardware design. So we implement it as a linear cyclic systolic array-based accelerator using small and very simple logic components, and this design helps us to maximize parallelism and minimize memory bandwidth and footprint. So processing core is the basic compute component, which computes the intermediate bit vectors, those 4 of them, along with the RD bit vector, which is the saddest bit vector. And when we add the flip-flop based storage logic around this processing core, we define a processing element, PE. And multiple PEs are concatenated to define a processing block. So we also have DC SRAM, which stores the reference text, the pattern bit mass for the query read and the intermediate data generated from PEs. And we also have TB SRAMs, which store the intermediate bit vectors generated by each PE of GenASM-DC, which will be later used by GenASM-TB. And we implement GenASM-TB hardware using very simple logic again, which reads the bit vectors from one of the TB SRAMs using the computed address, and then performs the required bitwise comparisons to find the traceback output for the current position, and finally computes the next TB SRAM address to read the new set of bit vectors. And after GenASM-TB finds the complete traceback output, which is also called stigar string, it writes the output to main memory and completes its execution. So we demonstrate the efficiency and flexibility of GenASM by describing 3 use cases of approximate string matching in genome sequence analysis, and read alignment and pre-alignment filtering are coming from the read mapping pipeline, as I have introduced as part of the background material. And as a third use case, which is edit distance calculation, this is a fundamental or very fundamental operation in genomics that measures the similarity or distance between two arbitrary length sequences. So this is, as I said, Bitap is inherently calculating edit distance. So that's why it is a very natural use case for GenASM. And we believe GenASM Framework can be useful for many other use cases, and we discussed some of them briefly in our paper. So we evaluated these three, and we discussed some of other use cases, including generic text search, for example. Let's look at our evaluation of the GenASM design and framework. So we evaluate GenASM using a combination of synthesis of our DC and TB accelerator data paths and a detailed simulation-based performance modeling. And we evaluated 16 gigabytes of HMC-like 3D stacked DRAM architecture with 32 walls. So our accelerators can be ported anywhere in the system, but in order to achieve high parallelism and low power consumption, we place both DC and TB along with the associated SRAMs in the logic layer of 3D stacked memory. And as I will show next, our hardware is highly efficient in terms of both area and power. So they are within the budget limits of the logic layer. So for the use cases we evaluate, we compare GenASM with state-of-the-art software and hardware baseline. And as you can see here, for the read alignment, we compare with two commonly used tools, which are called Minimap2 and BWA-MAM. And as hardware baselines, we compared against GACT of Darwin, which is for long read alignment, and ScyllaX of GenX for short read alignment. And for pre-alignment filtering, we compared against an in-house filter from developed by Mohammed, Suji. And for edit distance calculation as a software baseline, we compared against EDLab, which is an edit distance calculation library, software-based library, and an ASAP, which is a FPGA-based accelerator for edit distance calculation. So I won't go into details of the our evaluation methodology for each use case, but you can either refer to the slides later or our paper for more details. Okay, so we synthesize our DC and TB accelerator data paths with a typical 28 nanometer low power process, and both accelerators operate at 1 gigahertz. We find that for 32 GenASM accelerators, one for each HMC volt, total area overhead is 10.69 millimeters square, and total power consumption is 3.23 watts. So these may not make a lot of sense to you. So to give a comparison, we observe that both area and power consumption of GenASM at a single volt is around 1% of the area and power consumption of a single Xeon CPU core. So we find out that GenASM has low area and power overheads, which is important. So let's look at our results for the first use case, read alignment. So as software baselines, as I said, we compare with BWA-MAM and Minimap2, and we find that GenASM achieves 648 times speed up over 12 thread runs of BWA-MAM and 116 times speed up over, again, 12 thread run of Minimap2, while reducing power consumption by 34 times and 37 times. This is for a software baseline comparison for long reads. And compared to the GACT of Darwin, which is the state-of-the-art, as I said, long read alignment accelerator, GenASM provides 3.9 times better throughput, 6.6 times better throughput per unit area, and 10.5 times better throughput per unit power. And again, this is for long reads. When we look at short reads compared to the same software baseline, scenario is similar. Again, GenASM provides significant speed up and reduction in power consumption. And compared to the ScyllaX of GenX, which was the hardware baseline for this use case, GenASM provides 1.9 times better throughput while using 63% less logic area and 82% less logic power. So as I said, GenASM is highly efficient in terms of both power consumption, and it has a very low area overhead, again, compared to even this hardware baseline, which are state-of-the-art. Let's look at our results for the second use case briefly. So here, I won't cover all the results in detail, but here's the key takeaway. Compared to Suji, which is, as I said, state-of-the-art FPGA-based filter, GenASM is more efficient in terms of both speed and power consumption while significantly improving the accuracy of pre-alignment filtering. So this is important because when you decrease the false accept rate, which is one metric for accuracy significantly, and as you can see here, GenASM is doing that, you can, which means that you are filtering a lot, so there will be less amount of alignments that needs to be performed in the downstream. So the combined execution time for pre-alignment filtering and the alignment step would be very would be reduced significantly, so that it would affect the overall execution for read mapping. So it is critical to improve the accuracy by decreasing the false accept rates. And briefly, let's look at our results for the third use case. Here, compared to the software baseline, which is EDLib, GenASM provides 2 to 4 orders of magnitude speed up while reducing power consumption by 2 orders of magnitude. And compared to the hardware baseline ASAP, GenASM provides 9.3 to 400 times speed up while consuming 67 times less power. So again, we find that GenASM is significantly efficient in both metrics, both speed up and power consumption, compared to the baselines for this use case. So before I conclude, here's a list of additional details that we have in the paper, including algorithm details, more detail about use cases, as I said, evaluation methodology details, and some other additional results for all these use cases, along with a dedicated section where we explain our sources of improvements in terms of algorithm level, hardware level, and technology level. So to conclude, in this work, we focus on approximate string matching because it is one of the main bottlenecks of genome sequence analysis. And we propose GenASM, Approximate String Matching Acceleration Framework, to accelerate multiple steps of genome sequence analysis. And it is the first work that enhances and accelerates Bitap for ASM with genomic sequences. And in GenASM, we co-design our modified, scalable and memory efficient algorithms with low power and area efficient hardware accelerators. And we evaluate 3 different use cases and show that GenASM is significantly more efficient for all the 3 use cases in terms of both throughput and throughput per unit power than state-of-the-art software and hardware baselines. So as I and Juan already said, this will appear in Micro 2020 next Wednesday. So I will present it next Wednesday, and you can find our preprint. It's not preprint online. You can find our paper online. So thank you.